Good afternoon, everybody. I'm very happy to be part of this symposium, and I think I probably have the easiest task because I'm going to talk to you about the pharmacology of opioids. And as far as we understand, the opioid receptor is the same in an English rat and an American rat and everything else. So the pharmacology is behind it, and yet as human beings, we operate in a kind of social setting, and as Bart has been pointing out, that even the pharmacological effects of opioids on the central nervous system will have implications for behavior of clinical population, individuals, etc. But this is the pharmacology. So we know extremely well that pain is a kind of multiple series of pathways within the central nervous system. We have our incoming peripheral information, activity, integration, amplification on many cases within the spinal cord, but modification. And so we all know that, in fact, a very important site of action of opioids is here at the spinal cord level, where incoming peripheral information can be modulated, inhibited by opioid receptors. But if that doesn't happen, the messages then go to the sensory areas of the brain, the thalamus and the cortex, the emotional parts of the brain, the limbic brain, probably generating the comorbidities that occur in individuals, and then descending controls linking back down again. So, as I kind of intimated, here at the spinal cord level, we have mechanisms of amplification, central sensitization, but equally a balance, inhibitory systems, of which opioid receptors are the key inhibitory system here. So they are able to modulate those incoming messages before they enter the central nervous system. And then again, particularly in the limbic brain, but also in sensory areas as well, there are opioid receptors that are able to operate on higher processing of these incoming events. So, opioids, morphine. We have families of opioid receptors. They've all been cloned, they've all been isolated, and we can look at the individual makeup of the amino acids. So this is a neural membrane. This is common motif, 7 areas that cross the membrane, and this is like this superfamily. Beta receptors, for example, for noradrenaline are like this, and so on and so on. So these are receptors where the binding of an opioid to the part on the outside of the membrane changes the conformation and leads to inhibition. And this is inhibition of neurons in terms of the analgesia, but there's also inhibition of smooth muscle. We find opioid receptors in the GI tract, so that the constipation, the effects on bladder function are smooth muscle as well. So these receptors are found centrally and peripherally. And the sequences are such that there are 4 opioid receptors that we know of, and these just appear to be common to all mammals, and probably pre-mammals as well. So, an inhibitory receptor. But what do they do, and where do they work? Well, very briefly, we know that out here in the periphery, if you have inflammatory pain, it's a chemical activation, neuropathic, it's ion channels, into the spinal cord, mechanisms of amplification, the cortical, the limbic systems, the changes in the monoamines, the comorbidities, but the use of antidepressants to control pain, and these important glutaminergic pathways. So we know that opioid receptors can inhibit here at the spinal cord level. They can also inhibit up here, particularly in the midbrain and the brainstem. And so we have 2 predominant roles of these receptors. One of them is a presynaptic inhibition of the release of transmitters, and we'll look at this in a moment. So the incoming transmitters like glutamate and peptides are opioid receptor activation, will inhibit the release of those transmitters here in the spinal cord. There'll be inhibitions in the brain and leading to a suppression of painful messages. So the 4, and the terms, they used to be called opiate receptors. Opioids are used, they're all synonymous, so I wouldn't worry. Opioids is probably more commonly used. But the mu receptor for morphine, the delta receptor for some of the enkephalins, and these are the natural opioid peptides working at these receptors. Most of the clinically used drugs, and morphine is the gold standard, work on this mu receptor. These are synthetic drugs that work on delta and kappa, and now there's drugs coming along that work on this ridiculously named, it used to be called the opioid receptor-like 1 receptor. It's now called nociceptin opioid peptide receptor, since that's the ligand. I can't see why they couldn't think of another Greek letter. But anyway, those are the 4 receptors. But the clinically used drugs predominantly work on this mu receptor. So just a brief word about the endogenous opioids, and I think my pointer's almost wearing out here. The roles of these are inhibitory, because they're working on these inhibitory receptors, and the receptors predominantly work by opium potassium channels in neurons and smooth muscle. You hyperpolarize, you inhibit function. The kappa might close calcium channels, which is again inhibitory. But what do the endogenous opioids do? Well, they are involved in suppressing pain, and one of the nicest examples is an activation of descending control, so inhibition, lovely work by the Hamburg group, inhibition of spinal cord by placebo. But this is naloxone reversible. So this is telling us, since there's no exogenous opioid given to these individuals, that placebo analgesia has utilized an endogenous opioid system that's triggered probably opioid receptors in these limbic parts of the brain because it caused a descending inhibition. So they have a physiological function, but their lifetime is short. They're nothing like as potent as the synthetic drugs like morphine. And so, under most circumstances, endogenous opioids have a minor role in controlling pain. So, where does it all start? Well, I think it was 1974, Tony Yaksh, who was a basic scientist in San Diego, slipped a catheter into the epidural space of a rat, gave morphine, and we saw an analgesia. And that was a really important study because at the time, the action of morphine was thought to be somewhere in the brain. He showed clearly there was an action at the spinal cord, and it took a year to translate that preclinical data into the use of the first epidural opioids in patients. And so at the spinal cord level, there are opioid receptors made in the dorsal root ganglion. So this is the cell body of peripheral nerves. Not only do they cause excitability, they have these inhibitory receptors that are transported to the central terminals of the A delta and C fibers. And if you do sort of rhizotomies, 75% of these receptors are presynaptic. So the bulk of the spinal action of opioids given directly, but with the same with a systemic drug, is to stop the release of transmitter into the spinal cord. And in neuropathic pain, there are indications from clinical and preclinical data that opioids are effective, but higher doses are needed. And that, I think, is explicable by the fact that since the receptors are made in the peripheral nerve, if you have a neuropathy and damage to that nerve, you may be making or transporting less opioid receptors. So, presynaptic opioid receptors, they're found in the small fibers, the A delta and the C. The large A beta fibers do not contain or make opioid receptors. So this allows opioids to control the input from the nociceptive pathways whilst leaving tactile sensitivity unaltered. But we know that peripheral activity can cause central hyperexcitability within those same synapses within the spinal cord. And the activity of the spinal cord neurons, if we don't presynaptically inhibit activity, is going to lead to increased excitability of the spinal cord neurons, increased messages up to the brain. So another mode of action of opioids, if you remember, 75% are presynaptic. The other 25% of the spinal receptors are actually on these spinal neurons. So the next slide sort of gives you an overview of this process. Here we have incoming C or A delta fiber activity. It could be from osteoarthritis, it could be from cancer, it could be from surgery, it could be from trauma, it could be from neuropathy, releasing glutamate and substance P onto their receptors. These presynaptic opioid receptors, if you just look at this, along comes the opioid, inhibition, hyperpolarization of the terminal, reduced transmitter release. And then the postsynaptic neurons here, inhibition of firing of the neurons. And so you have a kind of dual control of the output of the spinal cord due to the ability of opioids to control the input and the output functions. And all opioid receptors in some way or another seem to have this ability, but there's also the descending controls which we'll return to, and opioids can interact with those and produce another indirect inhibition of spinal output through activating descending inhibitory pathways. So if you ever had to build a nervous system from scratch and you decided where you're going to put opioid receptors, I think these are the best possible places for controlling pain. So these spinal mechanisms then, the spinal neurons receiving painful inputs, it's this block of transmitter release followed by the postsynaptic inhibition that would allow, at a suitable dose, opioids to almost completely suppress firing of neurons. And experimentally, we can show this. The issue, of course, is that those same opioid receptors are found peripherally and in other CNS areas, and so a maximum dose may not be achievable, a maximum suppression, because of side effects from other functions. But at the spinal cord, we have a pretty pure action on nociception, and that's just an overview of those activities. So in reality then, we have a block of the release of transmitters from C fiber terminals that then prevents incoming messages. It may not be a complete block, so any transmitter that's still released will activate the neurons, but then the postsynaptic inhibition adds to that. And if you look at the spinal cord here, the opioid receptors are on the top in yellow, and this location is on the terminals of these small fibers in the superficial spinal cord. So that's this spinal cord action. And this is quite a nice picture. And this is another one. These are not opioid receptors or transmitters. It's the NASA calendar for next year that you can download, but kind of looks a bit neuronal anyway. So that was the spinal cord action. What about this action up here in the brain? So you could theoretically target those spinal cord mechanisms with epidurals and intrathecal injections, for example, and this is done. But generally the drugs are given systemically, and so now there'll be access to the supraspinal opioid receptors up here. And we know that as we move from a kind of acute pain into a persistent pain, in the spinal cord, we can have central sensitization, adding to pain. The normal balance between descending inhibition and facilitation changes. We gain facilitation, we lose inhibition, and opioids working in these supraspinal sites are able to modulate this activity. But of course, another tactic is, since we're losing noradrenaline function, that drugs with a noradrenaline reuptake inhibition can equally work to bring back and restore these inhibitions. But in this circuitry, there are a number of ways to function: sensitization at the spinal cord enhancing, descending facilitation, descending inhibition, and opioid modulation at the spinal cord. So these are mechanisms which we know are pretty much directly at the spinal cord level, but they're subject to bidirectional controls from the brain, excitatory and inhibitory, and these can change as well. So when we think about the supraspinal actions of opioids, we have this central gray, this rostroventral medial medulla, we have inhibitions and excitations, we have off cells, which are the inhibitory side, on cells, descending facilitations. And what opioids up here do is to switch inhibitions on, they activate the off cells, they switch facilitations off. And so they add to inhibition of spinal activity through descending pathways as well. But there can be a payoff to that, and it relates to the fact that these synaptic events and the ability of opioids to pre- and postsynaptically inhibit isn't restricted to those incoming pain pathways or to the descending pathways, but that opioid receptors are found in many other brainstem areas. They're found out in the periphery, and alongside the analgesic actions, those opioid receptors can produce problematic side effects. So this is just a summary of the analgesia, the points that I made: pre- and postsynaptic inhibition at the spinal level, switch of through supraspinal opioid receptors to favor inhibition. But now down here with systemic opioids, there are other additional problems, and I'll return briefly to those. So if we've got problems with drugs like morphine and oxycodone, which are mu opioid agonists, and I think most of the clinically used drugs predominantly are mu opioids, what about the other opioid receptors? Well, delta, no drugs have come through. Kappa, drugs like pentazocine have been suggested to be kappa opioids, but the purer ones seem to have gender effects. They seem to work better in females, particularly with red hair, and that might relate to the gene that makes this receptor. Hallucinogenic side effects, kind of strange side, and this ORL1 receptor, maybe in some conditions can enhance pain, but there has been this description of this drug, sevronopidol, or something hard to pronounce, which is partly an ORL agonist as well as a generalized opioid agonist. And so we'll see what that pharmacology brings. The main difference, as far as I can see, between opioids is their dose, their potency, and predominantly their half-lives, for example. We know that codeine is a prodrug for morphine, and so variation in the ability of the enzyme that metabolizes codeine to morphine can account for much of the variability in the analgesic effects of codeine. We have the fentanyl-like drugs, fast onset. Heroin, of course, is another prodrug for morphine. Oxycodone, I think, a mu opioid, but it's been used in combination with naloxone. And then naloxone, an antagonist. And then buprenorphine. And I never really kind of fully can get to the bottom of the pharmacology of this agent, but it might be a partial agonist of the mu receptor, an antagonist of the kappa, the delta, agonist, etc. But it would appear that predominantly the biggest difference with buprenorphine is that it sticks to that mu opioid receptor for much longer and in a different way to many of these other drugs as well. And that might be important. So an ability to control excitability of nociceptive neurons. If there weren't side effects, I believe it probably could be 100% control, but the side effects intervene. So what do we have? Well, Morpheus was the Greek god of dreams. I kind of tell the medical students, oh, morphine's named after Morpheus, and they think it's the bloke in the Matrix, but well before that, the Greek god of dreams. It's a sedative. Respiratory depression in the brainstem. This is a reduction of sensitivity to PCO2. And the cough suppression, constipation, that's enteric opioid receptors. Nausea and vomiting, there we can control that with an antiemetic, but this is a brainstem action. And then dependence, and I haven't got time to talk about this. There clearly is a physical dependence with opioid receptor activation, but I believe that the psychological, the kind of craving for the drug might be partly offset by the presence of pain, so that opioids may well trigger reward and reinforcement and psychological dependence, but pain itself is aversive and unpleasant. So in the presence of pain, the triggering of the reward system by opioids may be less than with street use, where there isn't any pain at all. So, not side effects. Well, there is a relief of anxiety, so that could be useful pre-surgically, and certainly with street use of the opioids, people are not generally medicating for pain syndromes. They're going for that relief of anxiety, that feeling of being wrapped in cotton wool, your cares and worries have gone. If you have chest surgery or bowel surgery, the anti-cough and the constipation can be useful. And of course, opioids can be helpful in treating diarrhea as well. But those side effects are there, and because all of the drugs seem to be predominantly working on that same mu opioid receptor, I don't think we see an enormous variation in their prevalence either. So just a few kind of final words about some other issues about opioids. Opioids, if you kind of think about it, are the major class of drug used in pain that activate inhibitory systems. And so the general kind of premise here is that you have a level of activity in your nervous system without pain. In the presence of pain, activity increases in your sensory pathways. You have too much excitation, you have pain, and by titrating up an opioid, you can increase inhibition to balance that excitation. And if you think about all the drugs that work, opioids are the only pain drugs, really, that directly activate an inhibitory opioid system. Other drugs might increase inhibitions, but it's a little bit indirect. So this leads to a problem of opioid-induced hyperalgesia. So there's been a lot of discussion of this. It might well be a fact in some patients. It's clearly observed in humans and animals, and in fact, 2 weeks ago, a study of patients with high-dose opioids in the community with sensory testing revealed a hyperalgesia. And basically, what it seems to be is that the systems that are inhibited by opioids at the spinal cord level and the brain descending pathways start to, in a sense, fight back against the inhibition. So the opioids are inhibiting, the systems that are inhibited start to bring back their activity, possibly because for a central nervous system, too much inhibition might be fatal. So the brainstem and the spinal cord pathways are clearly altered. So if you go and look in the pathways, the opioid receptors are here in the brainstem and midbrain and the spinal cord. You see adaptive changes here in animal models of opioid-induced hyperalgesia, but the preclinical evidence, interestingly, suggested that pregabalin, working here, and dexmedetomidine, this noradrenaline drug, working here but mimicking descending controls, could attenuate opioid-induced hyperalgesia. And in fact, there's now at least 2 clinical studies suggesting this translates extremely well. So again, something to be wary of. And what about opioids on their own? Well, we know that pain is complex. Combinations of drugs with different targets can be helpful, and you may want to combine to increase analgesia, or simply reduce the doses of the 2 drugs, reduce side effects, and you might just go for an additive effect. And one of the best examples of this is morphine and gabapentin, the Ian Gilron paper. There have been some others subsequently. This was in cancer pain. But one of the issues here is that you could increase analgesia with the combination, but you also increase side effects. And so it's this kind of payoff between one thing and another. And as a consequence of combinations, I just want to kind of draw your attention to this drug tapentadol, because this is a mu opioid receptor agonist. It's a weak one, and it also pharmacologically increases noradrenaline levels through an NRI inhibition. And I think this is a nice example of a combination drug within a single molecule where, the opioid load of the drug, if I can find my talk, might actually be less, because at any given dose, there's a substantial noradrenergic component to its action. So compared to a pure mu opioid, the side effects appear to be less, and it seems to have efficacy in a number of different pains. So kind of going on into the future, Ralph Baron and the German Neuropathic Network in patients with neuropathic pain have described subgroups of patients. And this has been done in other pain groups, including patients with persistent post-surgical pain as well, this sensory profiling as a basis for subgroups. And I think what we don't know at the moment is with opioids, for example, and this shouldn't be too difficult to do, is that are there particular subgroups of patients where increasing inhibition through opioid receptor activation is particularly advantageous. And I think having an idea about subtypes of patients who might well respond to opioids better than others may prevent possibly some of the overuse that is being indicated. But they are extremely useful drugs. They have this pharmacology, and I'd just like to thank my research team, 2 of them here arriving this afternoon. Thank them for the work that we've done with opioids, and thank you to Bart for inviting me along this afternoon, and thank you all for coming. Thank you.